Continue with the chapter on the body fluid compartments. So now let's talk about the composition of the different fluid compartments. So we have intracellular fluid compartment over here, and then extracellular fluid compartment, which is divided into interstitial or ISF, and plasma or blood. So if you look at the concentration of different cations and anions in your blood and in your cells, in the ECF and in the ICF, there is a significant difference in the different concentrations of cations and anions and even sugars. And this is very important because you must be aware that that your cells have different functions, and the ECF is like the milieu interior. So the ions, they cannot be the same over here. So if we look at the cations like potassium, if you look at potassium, the intracellular fluid has a lot of potassium. It's heavily concentrated in your cell. It is the maximally concentrated cation at 140 milliequivalents per liter. An equivalent is like when you take the number of moles and divide it by the valency of that particular ion. So it applies to ions. And I just want you to remember that equivalent is just when you divide that the number of moles by valency. So potassium has a valency of 1, so you can divide it by 1. Okay, so then you have your ECF. In that, for potassium, you could see that the interstitial concentration is 4 and plasma is 4.2. Why is it that there is a slight difference in the concentration of a cation between plasma and interstitial fluid, even though potassium can freely communicate and cross the capillary endothelial membranes, well, between plasma and interstitial fluid? That's because of something called the Gibbs-Donnan effect, for which reason the plasma with its protein is able to attract more cations and kind of repel some of the anions, which I'll be speaking about in more detail in some time. So the sodium, if we look at it, in the cells is less. It's around 14 milliequivalents, while it's around 139 to 140 in the interstitial fluid compartment and 145 in the plasma. Now if we look at chloride is around 4 in the intracellular fluid compartment, and in the ISF it's 28 and plasma 24. This discrepancy again is due to the Gibbs-Donnan effect. And bicarb, you should remember this because it's for that acid-base balance. It helps regulate the pH. So if we see the intracellular fluid compartment, it's got 10 as its concentration, 28 for interstitial fluid, 24 for your plasma. Then some other important ions now: phosphate, HPO4 minus H2PO4. They constitute a very important part of the phosphate buffer system in your cell. So you have 11. This is the maximum concentrated anion. So this is maximally concentrated anion over here in your cell, inside your cells. When we look at the interstitial fluid compartment, it's 2, and it's also almost 2 in the plasma also. There's hardly any difference over there. Now the other things you should be aware of is protein. So protein is concentrated in your cells. Your cells have DNA, they make RNA, and that makes protein. So if we look at protein, we have 4 in terms of its concentration over there. Now if we look at ISF, it is around 0.2 and only 1.2 in the plasma when you compare it with protein in terms of moles over here, milliosmoles per liter. So milliosmoles per liter, right? Milliosmoles, you can even say milliequivalents in the case of these ions, but milliosmoles is based on the millimoles concentration over here. Okay, then we have calcium. Calcium is very, very low in your cells. It is 0.0002. It's in the nanomole type of level when it comes to your cells, because a little calcium can change the way your cells function. Calcium is very, very influential. When Otto Levi got the Nobel Prize for Physiology, he said the line, "Das Calcium est alle," means calcium is everything in physiology. This tells you how important calcium is to in terms of physiological, biological systems over here. you cannot imagine almost any biological process without the role of calcium being a very subtle but very important player in terms of biological processes. So, but when it comes to your interstitial fluid compartment, you have around 1.2 millimoles of calcium or 1.8 millimoles of calcium in the plasma over here. Can you see? So 1.2 in the interstitium, 1.8 in the ECF, in the blood over there. And then magnesium, you can also remember this. It's very important and underrated. People don't know how important it is. 20 millimoles in the intracellular fluid compartment, 0.7 in the interstitial fluid compartment, 1.5 in the ECF compartment over there. And why should you know about magnesium? Magnesium mimics the role of calcium sometimes. You see, calcium, like I said, it's very important in biological systems. One of the things it does is stabilizes the membrane of excitable tissue, like your neurons and your skeletal muscle and some of your smooth muscle. So in the absence of calcium, your neurons they tend to fire much easily, much quicker. Magnesium also helps in stabilizing your neurons, and magnesium is also enzyme factor for your electron transport chain. So if you have low magnesium, you don't make enough ATP in your cells and you get tired very quickly. And if you have low magnesium, you are prone to arrhythmias of your heart, because that's also an excitable tissue, and magnesium stabilizes the membrane. So that's why heart patients with low magnesium don't do so good compared to heart patients who are given magnesium supplementation. So if somebody's had a heart attack or some arrhythmias, sometimes a magnesium supplement would be recommended by a good cardiologist for these people to have a better prognosis and outcome of their disease. Then if we look at the osmolar activity, so the number of osmoles are around 281. If you see osmolar activity, that's also 281 over here in the interstitial fluid and 282 in the plasma. Now, so that means there's a little more osmolar particles, osmotically active particles in the plasma, in the blood, compared to in the interstitial fluid. Now, why does this happen? Again, this has something to do with Gibbs-Dunn. When it comes to differences between the plasma and the interstitial fluid, there is a concept called Gibbs-Dunn, which I'll be speaking about very soon. But just be aware of these discrepancies over there. And then we have your osmotic pressure. So if you look at osmosis, I'll be talking about osmotic pressure in the transport across membrane topic, which is the next thing we'll be doing. Here, I want you to know that when you put salts or solutes in a solution and you separate it from another solution via a semi-permeable membrane that allows only water to go through, that is the solvent to go through, then there is a sort of a pull that the salts have on the compartment with lesser salt, so it pulls water towards it. So you can measure it in terms of pressure, and I'll be elaborating on that more when we go to that chapter. But the point is, the osmotic pressure is around 5,423 in terms of millimeters mercury for this one, all these osmolar particles over here based on the osmolarities, and this is 281, so that's again 5423, and then we have 5443 over here. So there is a slight difference of around 17 to 20 millimeters mercury pressure, and I'll be explaining about that when we talk about osmosis in more detail over here. So this is the composition of your various fluid compartments, and you should be aware of it. Why should you be aware of it? Well, because if we look at these fluid compartments, now sometimes you should be aware that some fluids have more fluid compartments expand and some contract. And how do they expand and contract? We'll discuss about this soon. Certain diseases like heart disease or liver disease or even kidney disease can lead to an expansion of your extracellular fluid volume, and that can give rise to swelling in the body and edema, and even in the third spaces like the pericardium, peritoneum, and pleural spaces that fill up with fluid over there. So I'll be talking about this and a concept called Starling's forces very soon.